In the previous tutorial, we generated two datasets from Wikipedia. We filtered molecules based on the rule of 5 and flagged compounds containing pane substructures. In part 4, we are going to compute fingerprints and cluster molecules. To work with molecules in silico, we need to represent them as numbers such as fingerprints, because computers fundamentally understand numbers, not atoms, bonds, or molecular drawings, at least not directly. In chemoinformatics, a fingerprint is a digital representation of a molecule that often encodes the presence or absence of specific structural features into a binary vector, and one could mean a chemical group is present, and 0 would mean the group is absent. Binary fingerprints in this example are thus a way to encode the structure of a small molecule into a fixed length vector consisting only of zeros and ones. So far, what has been said is about fingerprints that belong to the MaxCS key family. But there are many other types. We will not go into that here. Here we just illustrate the so-called MaxCS key fingerprint. The molecule has a NH group but does not contain a bromine atom. Chemical structures are complex and hard for a computer to compare directly, so we turn each molecule into a fingerprint. Once we have fingerprints, we need a way to measure how similar two of these vectors are. The most common way to do this in cheminformatics is with the Tanimoto coefficient. A Tanimoto coefficient of 1 means the two molecules are very similar or identical with that specific fingerprint. And a Tanimoto coefficient of 0 would suggest no shared features. Tanimoto ranges from 0 to 1. This is just to illustrate what we have seen in the previous slides. On this slide, we highlight the importance of specifying which fingerprint was used when reporting a Tanimoto coefficient, since the similarity value can vary significantly depending on the fingerprint type. When we compare molecules using fingerprints, like MAC keys, we sometimes get similarity values that do not really reflect true chemical similarity. Many unrelated molecules can share these common traits just by chance. So a similarity score, like 0.4, might just be fingerprint noise, overlap that happens not because the molecules are actually similar, but because the fingerprint is too coarse to tell the difference. On this slide, we just illustrate this notion of fingerprint noise. Now that we can compare molecules using fingerprint-based similarity, we can take it a step further. Instead of comparing two compounds at a time, we can group many molecules based on how similar they are to each other. That's what clustering is all about. Clustering compounds with fingerprints involves grouping molecules based on their structural similarity, as measured by comparing their fingerprint vectors. But this can be very, very challenging, even more so for millions of compounds. One goal of clustering is to group a large number of compounds into smaller, more manageable families. The final result is that you have a set of distinct families where members within each family are quite similar to each other, but the different families themselves are clearly different from each other. Note that some compounds may not fit well into any family and remain as singletons, that is, individual compounds that are too different from all others to join any group at the selected threshold value. In DataWarrior, there are different types of fingerprints. If we open the antifungal dataset, we can then select chemistry in the menu bar, then select from chemical structure, and then calculate descriptor, and then select a circular fingerprint named scale spheres. Once this is done, you can, for example, draw a benzene ring as shown on the slide. You can filter molecules that contain this exact ring, or using the fragFP or scale spheres fingerprint, select molecules that contain groups similar to this ring. By adjusting the Tanimoto similarity threshold with the slider, you can control how strict the similarity search is. If we keep only the molecules that contain the benzene ring, we are left with approximately 113 molecules. Now we remove this structural filter and we will do clustering. To cluster molecules, we will select chemistry and the cluster compounds. We select the value of 0.8 and we use the scale spheres fingerprints. In DataWarrior, clustering is performed using a hierarchical algorithm which can become computationally intensive when applied to more than 10,000 compounds. Hierarchical clustering is computationally heavy because it requires comparing every molecule to every other molecule in the dataset. For example, if you have 10,000 compounds, the algorithm must compute close to 50 million pairwise similarity values. These comparisons are stored in a large matrix and repeatedly updated as the algorithm builds a tree-like structure of clusters. This process consumes a lot of memory and processing time, which makes it difficult to scale to very large datasets. Once this is done, you have two new columns, one named cluster numbers, that you can sort by clicking on the column name. Here you see how many molecules belong to a given cluster, and the other column is named as representative. In simple terms, the representative molecule of a cluster is the one that best captures or summarizes the chemical features of the entire cluster. If you click on the configure view, you can set the preferred chart type to bar chart. Then if you click the XY button, you cluster number for x-axis, and for Y you select unassigned. This way you can see which clusters have many members and how many singletons you have. You can then save this plot to a file if you want. Now there is another way to look at similarity in DataWarrior. You can compute a similarity map. To do that, you select chemistry and then analyze similarity. And on the new window, you select structure of smiles with the scale spheres, fingerprints, and you use the same parameters you see on this slide. It should be the default parameters. You can now see the map. The lines connect molecules that are sufficiently similar based on a chosen similarity threshold. You will not get the exact same map on your computer. The nodes will be distributed differently. But if you, for instance, select a group that contains 3 connected nodes, if you click on one of these 3 nodes, you will see in a small window the neighbor tree for this selected compound. This selected node will be in the center of the neighbor tree. Now, if you select these 3 dots with the mouse or the touchpad, they will also be selected in the window named structure of smiles, as you will see in the following slide. So on this slide, we see what we discussed in the previous slide, the 3 molecules that are connected and found similar on this map. That's it for today. Thanks for following along. There's still a lot more to explore. I'll be sharing more tutorials to continue our journey with DataWarrior.